This is a cross-section of the female reproductive tract, focusing on the ovary. This organ operates under a strict dual mandate. It must execute gametogenesis, the development and release of female gametes, while simultaneously functioning as a systemic steroidogenic endocrine gland to regulate the rest of the body. If we examine its spatial layout, the interior medulla consists of loose connective tissue and vascular supply routes. The biological heavy lifting occurs here, in the outer cortex, a region packed with highly cellular stroma and distinct spherical structures of varying sizes. Most human organs maintain a relatively static physical form. The heart pumps and the liver filters, but their gross cellular architecture remains constant. The ovary is entirely different. It physically dismantles and completely reconstructs its own microscopic architecture every single month. The fundamental biological unit driving this monthly reconstruction is the ovarian follicle. We will follow it from a state of fetal arrest through intense morphological stratification into an explosive mechanical rupture, and finally, its reincarnation as an entirely new endocrine structure. In the ovary, physical form strictly dictates chemical output. The hormones circulating in the bloodstream are a direct consequence of the microscopic shape the follicle assumes on any given day. Long before birth, in the first month of embryonic life, primordial germ cells migrate from the yolk sac to the developing gonads. There, they multiply into millions of oogonia. By the third month, these cells begin the first meiotic division, but they do not complete it. They arrest in prophase 1, entering a state of biological suspension as primary oocytes. Looking at a highly magnified cross-section, we can see the baseline architecture of a primordial follicle. A single large primary oocyte is wrapped tightly in a flat layer of epithelial cells. This entire unit is isolated from the surrounding stroma by a clear basal lamina. These resting follicles are immediately subjected to atresia, a continuous, aggressive process of apoptotic decay where phagocytic cells dismantle and absorb them. The scale of this decay is massive. In a 5-month-old fetus, the ovaries hold over 7 million follicles. By the time that individual reaches puberty, atresia has reduced the reserve to roughly 300,000. Over a typical reproductive lifespan of 30 to 40 years, the system will select only about 450 oocytes to reach full maturity and undergo ovulation. The resting ovarian reserve is a state of constant biological attrition. Millions of structures are built and subsequently destroyed so that a fraction of a percent can survive. Beginning at puberty, the pituitary gland releases follicle-stimulating hormone, or FSH. Each month, this hormone rescues a small cohort of primordial follicles from atresia and initiates their growth. Prompted by FSH, flat epithelial cells thicken into cuboidal shapes, creating a unilaminar primary follicle. Simultaneously, the oocyte secretes the zona pellucida, a thick extracellular glycoprotein matrix that later binds sperm. These cuboidal cells rapidly multiply into stratified layers, now reclassified as granulosa cells. Despite the thick zona pellucida barrier, the granulosa cells extend microscopic filopodia through the matrix to connect directly with the oocyte's microvilli. This establishes gap junctions, allowing direct chemical communication and nutrient transfer. This cellular proliferation constructs the physical factory floor for a powerful new endocrine gland, one that will soon control the hormonal state of the entire body. Moving deeper into the cortex, surrounding fibroblasts condense into two tissues, a fibrous theca externa and a vascular theca interna. Functioning as endocrine tissue, the theca interna produces the steroid precursor androstenedione. An isolating basement membrane means only the thecal layers access blood vessels. Thus, androstenedione must diffuse across this membrane into the avascular granulosa. There, FSH-stimulated aromatase converts it into estradiol, a potent estrogen. Once synthesized, this estrogen diffuses back out through the basement membrane, enters the capillary networks of the theca interna, and distributes throughout the systemic circulation. This two-cell, two-gonadotropin system functions entirely through spatial division. The theca supplies the raw material, and the granulosa supplies the enzyme. This physical boundary ensures steroidogenesis proceeds in a strictly regulated sequence. As the follicle matures, the granulosa cells begin secreting follicular fluid, a viscous mixture of hyaluronic acid, growth factors, and high concentrations of localized steroids. Small pockets of secreted fluid appear between granulosa layers, eventually merging into a massive central cavity, the antrum. This expansion pushes the oocyte to the periphery, tethered by the cumulus oophorus, while the corona radiata tightly grips the zona pellucida. By the end of its 90-day development cycle, this dominant pre-ovulatory Graafian follicle has expanded to a diameter of 2 centimeters or more. This structure is now so large that it visibly bulges against the outer tunica albuginea of the ovary. It has engineered itself into a highly pressurized mechanical system, waiting for a final biochemical trigger. In the days preceding ovulation, the dominant follicle secretes immense levels of estrogen. This peak concentration signals the hypothalamus and triggers a massive sudden surge of luteinizing hormone, or LH, from the pituitary gland. This LH surge forces the primary oocyte out of its decades-long fetal arrest. It rapidly completes meiosis I, splitting its chromosomes and becoming a secondary oocyte. Simultaneously, LH forces the granulosa cells to hypersecrete hyaluronin. Because this molecule is highly hydrophilic, it rapidly draws water into the antrum, causing the internal hydrostatic pressure to spike. As internal pressure builds, the exterior wall fails. Activated plasmin degrades the collagen network, critically weakening the outer ovarian wall at a pale ischemic stigma. Next, prostaglandins diffusing through the follicular fluid reach the fibrous theca externa, triggering sudden smooth muscle contractions around the base of the follicle. Under extreme internal pressure and active muscular compression, the weakened stigma ruptures. The shear force violently expels the secondary oocyte, the corona radiata, and the follicular fluid out of the ovary and into the peritoneal space. Ovulation is a biochemically coordinated, structurally destructive mechanical event. The follicle effectively detonates to release the gamete. Following the rupture, the remnants of the dominant follicle do not die. Instead, under the continuing influence of LH, they undergo an immediate architectural reorganization. As we can observe here, the depleted follicle walls collapse and fold inward. Disrupted capillaries leak into the former antrum, forming a central blood clot, while new blood vessels rapidly invade the granulosa layer. The granulosa and theca cells undergo luteinization. They experience massive hypertrophy, increasing greatly in size without dividing, and transform into specialized lutein cells. This newly formed, highly vascularized endocrine gland is the corpus luteum. Its primary function shifts away from estrogen synthesis. It begins producing immense quantities of progesterone, the steroid hormone required to prepare the uterine endometrium for embryonic implantation. If fertilization does not occur, the corpus luteum functions for 10 to 12 days before undergoing apoptosis and leaving a dense connective tissue scar called the corpus albicans. If a pregnancy does occur, human chorionic gonadotropin, HCG, secreted by the embryo will sustain the gland for months. The 28-day ovarian cycle demonstrates that cellular structure dictates systemic function. The ovary constantly destroys and rebuilds its own microscopic architecture to manage the endocrine states that sustain human life.